Plants and animals share structural similarities and differences both externally and internally. In plants, these internal structures are adapted to diverse environments and are crucial for their survival and growth. The study of these internal structures is called plant anatomy. Different organs in plants such as roots, stems, and leaves show variations in their internal makeup. Notably, monocots and dicots are anatomically distinct, which is reflected in their tissue arrangements and growth patterns. Understanding plant anatomy helps us appreciate how plants function, adapt, and thrive in various conditions. As we explore further, we'll see how tissues and tissue systems form the foundation of plant structure, leading to specialized functions and growth. Plant tissues are groups of cells with a common origin and function. They are classified into two main types: meristematic and permanent tissues. Meristematic tissues are regions of active cell division, responsible for growth and found in areas like root and shoot tips. These can be further divided based on origin and location: promeristem, primary meristem, and secondary meristem, as well as apical, intercalary, and lateral meristem. Permanent tissues, on the other hand, are composed of mature cells that have lost the ability to divide. These include simple tissues like Parenchyma, Collenchyma, and Sclerenchyma, each serving specific roles such as storage, support, and mechanical strength. Complex permanent tissues like Xylem and Phloem are essential for transport and support within the plant. Higher plants, tissues work together as systems to perform specific functions. The three main tissue systems are epidermal, ground, and vascular. The epidermal system forms the plant's outer covering, including structures like epidermis, stomata, root hairs, and trichomes, which protect and regulate water loss. The ground tissue system makes up the bulk of the plant, consisting mainly of Parenchyma, Collenchyma, and Sclerenchyma, and is divided into cortex, pericycle, and pith. Vascular tissue system comprises xylem and phloem, responsible for transporting water, minerals, and food. Vascular bundles, which combine xylem and phloem, are classified as radial or conjoint, with further distinctions based on the presence of cambium. These arrangements differ between roots, stems, and leaves, and between monocots and dicots, reflecting their unique anatomical adaptations. Internal structure of roots, stems, and leaves varies between dicotyledonous and monocotyledonous plants. Dicot roots typically have fewer xylem bundles and a small pith, while monocot roots feature many xylem bundles and a large, well-developed pith. Stems also differ. Dicot stems have vascular bundles arranged in a ring and a differentiated cortex, while monocot stems have scattered vascular bundles and undifferentiated ground tissue. Leaves are categorized as dorsiventral dicots or isobilateral monocots, with differences in mesophyll structure and stomatal arrangement. These anatomical features are adaptations to their respective environments and functions, ensuring efficient transport, support, and photosynthesis. Secondary growth is a process that increases the thickness or girth of plant organs, mainly in dicotyledonous roots and stems. This growth is driven by lateral meristems, specifically the vascular cambium and cork cambium. The vascular cambium forms a ring and produces secondary xylem, wood, and secondary phloem, leading to the formation of annual rings that can be used to estimate a tree's age. Cork cambium produces protective layers like cork and secondary cortex, replacing the epidermis as the plant grows. Secondary growth provides mechanical strength, durability, and increased capacity for water and nutrient transport, which are essential for supporting larger and older plants. Monocots generally lack secondary growth due to the absence of cambium.